I don't think I would be exaggerating if I were to say to you that the Knight campus will be the single most transformative event in the history of the University of Oregon since its founding. Members of our faculty dreamt up this moonshot idea in my very first few months as president of the university. These researchers wanted a way for their basic science discoveries, the discoveries in the lab, they wanted to move those discoveries more quickly into testing and becoming cures and solutions to the problems facing our society. In turn, that market signal or those impacts would turn around and benefit and inform their basic science. We call this the impact cycle. One of the overarching goals of the Knight campus is to create a research-rich environment, gather together smart and innovative people, remove barriers, then that in turn would rapidly speed this impact cycle. We want to encourage collaboration and discovery and allow for the development of more life-changing innovations that will improve our world. When we got the largest gift in the history of public universities from Penny and Phil, I asked around who would be the midwife for this great experiment, the Knight campus. In a remarkable show of unity, especially for a university that doesn't have a surfeit of consensus, right? Everyone pointed to one man, our lecturer tonight, Patrick Phillips. Patrick's exceptional scholarship, his remarkable teaching, and his extraordinary leadership caused me to ask him to be acting executive director of the campus. Patrick has once again sacrificed his own personal and professional objectives to take on the extraordinary task of taking this kernel of an idea and transforming it into a reality. As the acting executive director of the Knight campus, Patrick Phillips is responsible for motivating and guiding a number of teams establishing the Knight campus for future success, including leading the design and construction process for new scientific research buildings, which will be built on the north side of Franklin, establishing governance and educational policies for the campus, and initiating and supporting innovative graduate internship and entrepreneurship programs. But first, and most importantly and always, Patrick is a professor in the Department of Biology and a member of the Institute of Ecology and Evolution. He has previously served as the director of that institute, the head of the Department of Biology, an associate vice president for research, and of course is now acting executive director of the Knight campus. He is a nationally recognized expert in ecology and evolution, and his research focuses on the biology of aging, genomics, and the genetics of complex traits. He has helped pioneer our understanding of how complex genetic interactions shape the way that organisms develop, behave, and evolve. And he is now applying these approaches during his free time toward discovering how to increase people's health spans and decrease the impacts of environmental stress over the lifetime of an individual. I am absolutely delighted to introduce to you my friend, my colleague, and the leader of our Knight campus, Patrick Phillips.
Thank you, and thank you all for coming out tonight. This is a great opportunity for the university to interact with the community, and it's a harbinger of many more things to come that are going to be great. And I think members of the community know that have probably heard rumors that President Schill is just a bundle of positive energy and engagement. And I'm here to tell you that that's 100% authentic. He's doing a great job, and it's been wonderful. One of the best parts about my current role is was driving around in the backs of a taxi cab in New York City as we're unrolling the announcement for the new night campus, and being able to see him in his full glory of just throwing out energy and directing things. It's been really exciting. So my goal here tonight is, I have a couple of goals actually. I want to tell you just a bit about the night campus, although the president already gave a pretty good introduction to that, so I won't dwell on that. I want to talk a little bit about my own personal development as a scientist, and just briefly touch on that, because what I'm doing now is not what I started out to do, and I think that that's also a story of the night campus that is illustrative. And then, of course, I mostly want to focus on the topic of the evening, which is about the biology of aging and where we stand, because I think we actually stand at the precipice of something brand new for our society, and we really should be thinking about it. And that's why I'm really grateful that you're all here to start a conversation, not just to hear me say things, but to really think about what the future is. And I chose this picture for the beginning here. This is the Egyptian god Anubis, who is the god of death, you may know. And at the moment of death, of course, you've appropriately had your heart removed ahead of time, I hope, as the Egyptians would do. And Anubis weighs your heart against the feather of truth. And depending on how that balance goes, determines on where you go in the afterlife. And so I think it highlights the fact that this balance between life and death is something that's been central to all cultures in humanity, and something that we want to do. I think it also points to the fact that I really hope the next Indiana Jones movie seeks the feather of truth, because I think it's something that we could really use at this moment moving forward. So a big theme of everything that we're doing, and as we think about starting a brand new operation at the University of Oregon, is really thinking about the interaction between these three components: science, technology, and society. And so science is a process in which we try to understand how the world works. We like to think that it's getting at truth with a capital T. That's a philosophical debate, but it is an important way of doing things. It's not the same thing as technology, and so the fact that iPhones exist is not science, but technology is often the implementation of scientific understanding. And a really important component of everything that we do in science in the modern world is the interaction between science and technology. Most of what we now do in science is impossible without advances in technology. Most of what's done in technology is not possible without scientific insights. And then finally, we have society. So I think it's fair to say that science and technology have been the major drivers of the societal progress, if you want to call it that, or changes at least, that we've seen over the last 200 years. I think for many thousands of years, we lived a very different kind of cultural life together, but the advent of the Industrial Revolution, and now we see in the technological revolution that we've experienced over the last 50 years, that we live in a very different world than we did before. And so understanding the interaction of all these things is really critically important. But before I get started, well, actually, I want to then highlight the fact that these are elements that come together in the night campus. And so this is the mission statement of the night campus. It's science advancing society. It's an outward-looking, facing component of the University of Oregon that is new to us. And the intention is really to, as the president said, to take discoveries that we've already been making, and instead of letting them rot in a scientific paper somewhere, figure out how to bring them into the world so that they can have an impact. And that could be discoveries about new health things. It could be policy. It could be a wide variety of impacts. Our most immediate need is to build these new buildings, and we're We're in the process of doing that. I think the members of the community, that's the first thing that you'll see. We're hoping to break ground in the fall. You'll probably see the first sort of construction starting in the spring of next year. And so it's going to be an exciting new change, a new shape to the skyline of Eugene. I think it's going to be a wonderful thing. It's going to be a new entrance along Franklin Boulevard to the entire community as well as to the university. So to turn now to the science, I have to actually start with my lab. And so everything that I do is really built on the work that the members of my lab do. And this is, again, a night campus theme, is that even as we hire maybe 30 new faculty, the multiplier of the impacts on students and the staff, so when the night campus is fully operational, we'll have about 700 new members of our community here, be generating on an ongoing basis about $75 million in new economic activity in the community. So this is not a minor sort of thing that we're getting started. And so this is partially why I'm a scientist, is because I like to be a teacher. Mostly, though, I'm a scientist because I'm a student. And I like interacting with other students because I like to learn new things all the time. And so this is a great community. So as I talk about the various work that we're doing, I'm not going to highlight the individual contributions that the members of my lab have done. Some of them are out in the audience right now. Just know that I love you and that it's everything that we've ever done is built on everybody's hard work and intellectual contributions. So I just want you to take a second. It's dark enough now, but I just want you to take a breather and really think about yourself for a second. Not in this egotistical way, but actually think about you as a biological organism, how you work, what's going on with you right now. I think we're obviously born into this world. We just go about our business. We interact with each other. It's very rare that we understand, and it's only in the last 50 years or so that we really understand how we function. And it's really extraordinary. Matter of fact, it's so extraordinary, I don't understand why we aren't giant puddles of goo on the ground, because it's a very complex set of things. And there's two elements that make us incredibly extraordinary as organisms. The first, and the central component is that we're made up of semi-autonomous cells, maybe 50 trillion. This is a developing frog embryo that's going through its paces. So these cells each have a copy of our DNA in it, but there's no central organizing unit that says it's time to put this cell here and that cell there. Somehow these cells all have to interact and communicate with each other and figure out how to make the whole out of these 50 trillion parts. It is completely mind-boggling that somehow this happens. Again, there's no central processor. When we think about complex engineering problems, we imagine someone designing how it's going to work. This is a self-organizing system, and all of life is this kind of process, self-organization. And the way that that works is through communication among the different components. The other extraordinary thing that animals have, in particular, is another kind of organization. So this is the view of just a very small section of a mouse brain, and each of the little shiny dots there is a neuron. So the other thing that is really extraordinary about us is that we think that we exist as single entities. That is, that we can say I or you, right? We have this sense. If I asked you to think about yourself right now, you could do that. But that knowledge is, again, made up of billions and billions, maybe 100 billion different neural processors, cells, neurons that are all interacting with one another, and somehow they get together and make us a whole that feels coherent to ourselves. As a matter of fact, we feel so coherent that we completely take it for granted. We cannot imagine anything else could possibly be true. But again, we are all these independent units. We try and say, well, the central thing is the brain. Well, the brain is not even a central thing. It's made up of all these little components that all have to interact with one another. It is completely mind-boggling to me that this works at all. And really, in science, we're just figuring out how this works. So I'm going to talk about aging tonight. And really, the process of aging is the breakdown of this process of self. It's how we slowly sort of lose this component of interactions both within cells or within our own brains, so that we lose that coherence over time. And so really the secret to combating aging is somehow to combat that process. So what is aging? This is my friend Charles Darwin, who's important in my life, although I've never met him. Again, these are the kinds of things that we tend to take for granted. Why even question? Well, of course, people get old. That's just what happens, right? And somehow we change as we get old. That's just what happens. But it is actually a phenomena. And what I want to start off with is just to take you through what aging actually means. And you think that aging and getting old are the same thing, but aging actually has a technical definition. So why do we care about aging? We don't need to worry about any of these details. These graphs show the rate of cardiovascular disease, dementia, and cancer. Okay, and on the bottom axis is age, and it starts depending on where you are. In cardiovascular disease at 16, for dementia, there's essentially no dementia below age 60, and for cancer, even children can get cancer, but it goes up. And what you'll see is overall, there's this phenomena that these things tend to go on an accelerating cycle as people get older. So we tend to talk about these as diseases of aging. But if you flip this on its head and thought about age as the causal factor, age is in fact the #1 causal agent of disease that we have in the Western world, as we'll talk about in a second. Okay? So aging is something different than just getting old. Something is changing such that these rates are accelerating. That's not the way it used to be for humans, and that's because we didn't live as long, right? So here are the major causes of death in so-called third world countries. So countries that tend to have low levels of income and social support systems. So overall, what you'll see is the top is like lower respiratory, so that's basically pneumonia, and in this case, it's probably mostly in children. Heart disease, AIDS, stroke, tuberculosis, malaria, premature birth weight. So many of these things are diseases of malnutrition and external diseases caused by infectious agents. And probably for most of our evolutionary history, this is what's dominated the way that we've lived. That's no longer the case. In the Western world, we have different kinds of diseases. And so people are dying of different things: heart disease, stroke, cancer, Alzheimer's disease and dementia, more cancers, diabetes, more cancer. You're getting a theme here. And so something has changed about why we're dying. So this boundary between life and death in the Western world versus traditional. And this really becomes stark when you actually look at longevities across the world now. So the countries that are in the reddish colors have a low longevity. So the ones that are in very deep red, almost black, the average lifespan in those countries is under 45 years old. Okay? On the other scale are green countries, and the darker green they get, like in the European and Scandinavian countries, the average longevity is over 80 years old. So even in the world today, we have a 40-year span of age disparity. And there's many things that could be said. I'm a scientist, not a social scientist or an economist, but I think this disparity actually begs the question about what's going on. But we do see that, that this is something stark. So something has changed in what we call the Western world or first world countries such that people are living much longer. And we can actually look at this directly. So along the axis here, we start at 1840 and end at 2010. And what you see is that life expectancy is starting similar to some of these African countries around 45 years old in 1840. And now we're well above 80 years old. And each one of the different lines is a different country like France, Germany, the US, or Sweden. Okay? So what we see is in the last 100 years or so, we've seen just a continuous increase in life expectancy. And right now, it doesn't look like it's sort of stopping, although there's some debate about whether or not we've hit the limit. So we'll talk about that. Okay, so this is a very recent phenomenon, and I think the thing to think about is all of this is going on. This is a huge demographic pressure that's occurring in our society that we're not really confronting or talking about in a very serious way. And I think we're still working on the social structures that were around in the 1800s for the most part, and even though the world that we live in is quite different. And so this demography is creating a destiny that we really need to confront. And what I'm going to be talking about tonight is changing that even more. And so we really need to think about this as a broader society and as our community. So what is aging? I keep talking about this. Well, let's look at it. So what this graph shows is it takes the probability of living to be 100 years old, and then it extrapolates it back, except it makes one assumption. And the assumption that it makes is that the probability of dying every year is exactly equal to one another until you reach 100 years old. So what do you see about this? So this is called constant mortality. Well, if we live this way, people would be sort of living to be about 20 years old. Okay, that's not what we see. It may be back in the caveman days that was going on. You know, the probability of being eaten by a saber-toothed tiger was about equal every year, and so maybe longevity was about like that. Certainly, longevities historically over human evolution have been quite short, probably more like that. But that's not what we see. So this is what the survivorship curves look like in the United States now. So you see that, as you know, men don't live quite as long as women, but there's not a huge disparity. So what do we see instead? We don't see constant mortality. Instead, we see very, very, very low mortality in the Western world early in life, and then a huge acceleration in mortality later in life. So what are the implications of that? Well, if we could stop time and whatever's going on biologically at age 60 and just control that rate of mortality, we would actually live to be 270 years old, okay? If we stopped it at age 30, we would live to be almost 2,000 years old, okay? It's unbelievable when you actually look at the numbers. And you say, well, that's insane, that's crazy, that couldn't be possibly true. We could never achieve that. Well, I would say there are people on this earth right now that live at that mortality rate. They just happen to be 30 years old, okay? So what the secret to longevity is not very profound. It's what's going on in a 30-year-old versus what's going on in a 70-year-old. That's it. That's the end of the story. You now know what the answer is, okay? Something has changed in us around 60 years old, and sorry for those of you who are around 60 years old, but something changes and the rate of mortality greatly, greatly increases, okay? My boss, too removed, is sitting right down here, so I can't insult him too much. This process is called senescence. And so aging is not just getting old, it's the accelerating rate of decline with age. Okay, so that's what aging is. So if we're going to sort of solve aging, the question is how do we change that rate of decline? And as I pointed out, even a slight deflection of that has a huge impact. Hundreds of years. And so it's not that far-fetched to think that we could change this in the near term. All right, there we go. And in fact, we know organisms that have done this, right? So this is a bristlecone pine. The oldest bristlecone pine that's known lived to be about 4,800 years. How do we know that? Count the rings. So what happened? So a graduate student from North Carolina in 1964 drilled into a tree and their drill got stuck, okay? And so they got permission from the Forest Service to cut the tree so that they could get their drill out. And it turned out they cut down the oldest organism that's ever been observed. So, and made it easy to count the rings. That was a mistake. But there are other organisms that are on this planet right now that sort of ask the question, what is aging? Is it a predetermined factor, or is there something biological that we can change? So a great example are actually in rodents. So we have a metal vole that my cat likes to catch. And they only live to be about 1 to 2 months old. And that's sort of what we think is, certainly a fast-lived rodent. But there's this attractive fellow at the top, which is the naked mole rat. They live in social colonies, and they live to be 20 years old. So these are both rodents. Somehow something biologically has changed that one can live many orders of magnitudes greater. So what's going on? What's happening in us? So this is a painting called The Fall of the Rebel Angels by Peter Bruegel. And I think this basically describes what's happening. So we have all these processes that are present in us, and somehow there's a battle of our greater angels, right? So at 30 years old, everybody's copacetic. 60 years old, something arises and something bad happens. So what we do know is that it's possible to break our functions and then to have accelerated aging. So this is Werner's syndrome, which is a breaking of a DNA repair system, and it leads to very rapid aging. It looks like this woman is just getting very old quickly. So at 56, she looks like she's in her 90s. Okay. Here's another even more famous example. You maybe have seen this. This is progeria, which is a breakdown of the envelope that surrounds the cell nucleus. These are kids, okay? They're only like 7 or 8 years old. So we know that it's possible to accelerate aging, but that's not going to get us to addressing the problem of aging. And so this kind of information accumulated, and then a revolution occurred in the early 1990s, late 1980s. And it occurred in the most humble way possible. It was a discovery that was made in a little nematode worm. And this worm is called Caenorhabditis elegans. It happens to be the system that I study. And what happened is that people started thinking about, well, can we actually study aging as a genetic process? It was thought that this would be impossible, that there couldn't be any single thing that could somehow address aging. But a couple of biologists started doing this, and in particular, they found a mutation that rather than making things age faster, it made them live longer. And this was the first time this had ever been observed. So this is how it works. So this is the way a worm lives. It turns out, so rather than years, they live in days, which is convenient from an experimental point of view. It makes them a little bit different than humans, obviously, in many respects. So a worm will live about, close to 20 days on average. And a very old worm, long-lived worm, is almost 40 days old. So this one mutation completely shifted the picture and made the worms live twice as long. So a single genetic change made them live twice as long. And since then, there's been other discoveries that can, in combination, can make them live 10 times longer. So if you think about it, you can imagine if we could make a single genetic change in you, or a couple genetic changes, and you would live to be 900 years old, just like that. That's a pretty profound difference, right? So it turned out that people cared about that, and they started spending a lot of money on it, okay? So there's been a huge amount of research in the last 25 years in this field, okay? So I'll just tell you a little bit about this one particular system, just to give you an illustration of how it works. So it turns out that this gene that was mutated is an insulin-like receptor, okay? So the way it works is that insulin is outside the cell, and when is insulin around? When you have food available, right? So there's food available there. When that happens, it then triggers a set of signals that come and they block this thing called a transcription factor. What is transcription factor? Its job is to turn on, it's a protein that turns on other genes, okay? So when there's food around, this transcription factor is blocked. It doesn't do anything. But when the insulin disappears, okay, then this signal goes away, and the transcription factor is released, and then it binds to DNA. And then we know what the genes are that it upregulates, it's called. It upregulates things related to stress. So the worm thinks that it's starving, and so it knows that it's in for the heavy weather here, and so it upregulates all these stress response genes. So it turns out by inappropriately upregulating the stress response genes, you can make a worm live longer. So that's cool. The amazing thing is that system is conserved across all animals, including humans and mice and monkeys and everybody else. And so this was a fundamental biological process that we didn't know about, that was discovered through genetics, and then allows us to then manipulate how stress response is turned on. And of course, it turns out to be incredibly complex, and many hundreds of people are trying to figure out what's going on. Okay, so that's the background. So what's my story? I actually started off a little different. So we could have titled this talk, Aging Biologist, Aging Biologist. So I started off as someone who was very interested in genetics, and especially using mathematical approaches to understand how organisms become different from one another, both within populations and between populations. So that's a genetic field called quantitative genetics, and it also is an important part of evolutionary genetics. So that's what I did. My first research, well, that will be that. I think I hit the wrong button. There we go. So my first work actually was on frogs. Frogs are an amazing system. We sort of scooted over that, but they go from egg to tadpole and so on and so forth. So I was very fascinated by those complex relationships with one another. But I quickly discovered that frogs are a horrible organism to study from a genetic point of view. They're just really hard to study. And in particular, I wanted to be able to study evolution in action. I wanted to be able to observe it as it occurred. And so I started working on this worm that a lot of other people were studying. It was actually the first organism to have its genome sequenced. We know as much about it as almost any animal. Its major competitor would be the ubiquitous fruit fly in terms of what biologists study. Okay. So I started using an approach which was to actually do selection experiments in the lab. And this is a field that's called experimental evolution. It sounds a little ominous, I guess. But the idea is to expose organisms to novel environments and then see how they change. And so the environment that I wanted to investigate, we've done many different kinds of projects this way, but one of them was to look at stress. So I wanted to select for different stress responses and then see how the organism changed over time, and that would help me understand the genetics of what the differences between individuals were in terms of what makes them stress resistant. And one of the major theories of differences in longevity between individuals, as we've already talked about, is the ability to respond to environmental stresses. And so the stress that we chose was heat stress. And so we exposed worms to very intense heat stress that would tend to kill them, and then see what happens. So we collected worms from nature, brought them into the lab, and then periodically exposed them to a high stress. So one interesting thing about these worms is if you raise them at a cool temperature and then expose them to stress, they die. So about 80% of them would die. But if you pre-expose them to a hot temperature that's not lethal, they actually do a lot better. Okay, so about only 20% of them die. So there is something about being pre-exposed to stress. So a little bit of, what doesn't kill you makes you stronger kind of thing. So then we selected them for a number of generations to see if we could increase their stress resistance, and that's what we did. So now even the guys who are in the cold environment are more resistant than the guys who have been exposed to stress. So we thought, aha, we've now figured this out. We now know how to make worms stress resistant all the time. And so now we're going to go in and look at the genes and then be famous, okay, to discover how to upregulate stress resistance. So we've done that, but we're not going to be famous. So it turns out that when we did this, so this is the genome of a worm, and we can use now modern technology to sequence the whole worm's genome and figure out every piece of DNA that they have. And then we can put it and ask which parts of the genome change as we did selection. And it turns out thousands and thousands of sites in the genome change. It's not one mutation. It's many, many places in the genome that makes one worm more resistant to another, and then we can accumulate those over many generations. So there isn't a simple answer in our case. There isn't a single gene that we can point to and patent, for instance. And so that's that. So it's like, well, life is complex. That's the way it goes. So there we were. Around the same time that we were doing this, the National Institutes of Aging, which is part of the National Institutes of Health, started a major project using mice to ask how can we make mice live longer with the idea that we're going to make then humans. So they started a system of giving a lot of different compounds or drugs to mice to see how does it affect their longevity. Okay, so they started this project and they worked on it a while. So the first thing they discovered is this is not a good way to make mice live longer. Okay, so you see the little mice on the head there. So it turns out that mice in the lab would live for several years. So these are very long-term experiments. They're very hard to do. They're quite expensive because mice are very expensive to take care of. And so they studied a number of chemicals and they thought, oh, this is great. They found some that made the mice live longer. And then someone said, all of that was done with a single strain of mice. Okay, so when we look at this other mouse, it's a little bit different. We don't see the same effect. And with, oh God, now the National Institutes of Aging said, what do we do now? Do we go back and then test 100 different strains of mice? It's very expensive, it'll take forever. And they said, no, that's not what we're gonna do. We're gonna use this worm and we're gonna do that. And so it was a perfect opportunity. I was the only person in the world stupid enough to study natural variation in these things, in worms. And so the National Institutes of Aging came to me and as part of an application process and said, let's start this new project. Why does this matter? It turns out that this is a general phenomenon where the differences between individuals and their genetic backgrounds and everything about them affects their response. So these are the top medicines that are used in the United States today in terms of total money spent on them at any rate. And so every blue human there, so let's take Nexium. I won't ask you to self-identify if you take Nexium, but probably a lot of you do, or have some sort of reflux issue. So every blue human is a member of the population that needs some treatment in which that particular drug works. All the red ones are people who it does not help, okay? So you'll see for all the top 10 drugs that are used, the best you can do is about 25%, okay? So this is a big problem. It turns out that we're all different from one another, and that influences the way that we respond to drugs as well as everything else. So if you're trying to find a drug that's going to make you live longer and healthier lives, then we have to address this. We have to understand how the different genetic differences among individuals affect their responses. And so that's why we created this new program that's called the Cenorhabditis Intervention Testing Program. It's almost as easy to say as the Campus for Accelerating Scientific Impact. I've gotten very good at saying that, I got to say. And so it's actually a consortium of 3 labs, my lab, and then a lab in Monica Driscoll's lab at Rutgers, and Gordon Lithgow's lab at the Buck Institute for Aging, which is in Northern California. And part of the idea here is that, and this is a great theme that I don't have time to get into, is another problem with science is actually replicating science. It's really becoming a challenge. And aging turns out to be a very difficult thing to study. So the whole point here is that we're replicating these studies simultaneously as we do them. So the whole idea of this project is we bring in a whole bunch of different drugs and we give them to worms. It's not the kind of drugs you're thinking about in Eugene. There are different kinds of things that are supposed to make the worms live longer and healthier lives, okay? And so what we did is we took strains that have been collected for multiple species, actually, of worms in this case, from all over the world, and they're quite genetically diverse, and then we subject them to these different compounds. So I'm just going to show you a couple of results and the kinds of things that we see. This is our best hit so far. These are 6 different strains on the bottom from 2 different species. We don't need to wonk out too much on this. This is and on the axis is the 0 point is what the control is with no drug. And then everything above is how much more you live when you're given the drug. So if we look at our favorite strain, My16, which turns out to be quite druggable, they live about 75% longer when they're given thioflavin T, which is a kind of dye, actually. Okay, so what we find is that every strain that we treated lives longer, but some live a lot longer and some live just a little bit longer. Okay, so this general phenomena that genetic background matters is one of the results that we see. But this is still quite promising if we can make a 75% increase in longevity just by taking a compound from the external world. That's a great kind of insight to have. So we started to do more work on this, and one of the first things that we wanted to do is make use of the technology we have. So it's actually quite tedious to look at a worm and wait till it dies. You have to keep poking it, and are you not dead yet? So that's how it works. So we have a high throughput means that we can do, which is to put the worm plates that we study them on a flatbed scanner, just like you use to scan your documents. And then we can scan them once every 10 minutes or so, half an hour or so, and then we can ask, are you dead yet? No. Okay, so if they're still moving, then they're not dead. So we did that with IoT. It's a great way. We can move tens of thousands of worms here and really do great screens. So we worked on that. Just one problem. When we put the worms on the scanners, they all died. Okay, so thiot was actually poisonous when exposed on the scanners. So there's something that we don't totally understand yet that probably makes this dye makes them extra sensitive to UV light. Okay, so what have we discovered? So we actually discovered a vampire drug, right? So it makes you live longer, not forever, but longer, but you can't go out at night. I'm pretty sure that thing wasn't moving when I put it in my presentation. Okay, so but actually thiot works, but probably we think by binding negative protein accumulations, and so it binds to them. So it actually makes the worms a little colorful when it's done. So maybe this is the proper vampire for those of you who are aficionados. This is the shiny vampire version of the world. So anyways, so don't start taking thiot unless you want to not go out anymore. So I'm not recommending this as a thing. Here's another compound that we studied that has some pretty interesting results. So this is resveratrol. So resveratrol is a component of red wine. So people got very excited about this and made millions of dollars trying to sell it as a way of improving your health through drinking red wine. And what we found is it does help a little bit, maybe more like on the 20% range, which is still significant if you're going to live 20% longer. But it only works in one species, C. elegans, and not in the other, C. briggsii. Okay? So I've actually personally collected both of these species in France. So I don't think it's just a cultural difference about their preference for wine. But overall, it shows that there can be maybe important biological differences. And this is the kind of thing that we're working through. And I won't bore you with all the details, but basically we're just sort of systematically moving through this. And we hope over the next 5 years or so that we'll move through several hundred of these compounds, and then we'll understand which ones are doing the right kind of thing, and then be able to bring those to mouse trials and then eventually onto human trials. And that's part of, again, the Knight Campus idea is how do we spin out these basic discoveries into long-term impacts. So here's a summary of some of these things. And basically it turns out to be sort of a negative result is that we have some things that are positive. Most are either mixed or negative, but only one is what we call highly robust, that it's always positive regardless of genetic background. So this is going to be a challenge, and it actually takes a systematic, determined effort. And a lot of science is not actually sexy or exciting. It's just hard work that we just keep doing because it's interesting and we love doing it. Okay, so this is the kind of thing, and it's a lot of work, and my lab has been doing a lot of work on aging lately. So when I tell people about this, and we're trying to find compounds that are going to make people live longer, their number one response is, that sounds like a bad idea, okay? And people have a very personal response to this, and they think that actually making people live longer is maybe not what we want to be doing as a society. So I want to explore that just a little bit. So why do people think that? Well, it turns out that the number of older people in the world is growing rapidly. Okay? So this is the total number of people ages 65 to 85 in red and above 85 in gray. And you'll see, you have to look, that's 2060 at the end. So it turns out, unless you're from the future, this hasn't happened yet. So we're in the middle part of this. But what we project is that over time, the aging population is going to continue to grow. And again, this is 2014 is right here, 2060 at the end. And so right now we have about 15% of the population or so is above 65, but we're projecting in 2060 that will be 1/4 of the population will be over 65. Okay, why does this matter? Well, people have basically two concerns. One is economic and one is population impact, and both of those are highly legitimate concerns. So what we think is, well, do we want old people around who are taking Social Security? And in fact, in 1934, I think it was, when Social Security was created, only 5% of the population was greater than 65. So the economics of that is really quite relevant. And it becomes more relevant in our state when we talk about PERS and all these other things that are driving a lot of our budget burden. The other one is in terms of overall population impact. So there's a lot of disparities again, or interesting differences in longevity. Again, in Europe, most of Western Europe lives to be in their late 80s. In Eastern Europe, they're still in their 70s as longevities. And so we know that there are large environmental effects, and I don't have time to get into those. In the United States, there's also large differences across the country. It's interesting to think about correlating this with electoral votes or something like that. It's actually a pretty good correlation. But overall, we know that these things are driving it. I just want to quickly just talk about the population growth part of it, because that's totally legitimate. We obviously are creating a load on this planet that we need to be attentive to. I don't think that that's actually over the long term a major concern. So this plots the rate of population increase versus child mortality, which seems like an interesting thing to do. You would think that as child mortality goes up, then population growth should come down. But in fact, it's exactly the opposite. So I'm going to animate this through so you can see this is starting in 1970, and I'm going to show you the next 40 years or so of what happens. You'll see a couple blips come through. It's Cambodia and Rwanda, and their mortality goes up due to some genocide that was going on in those countries. So this is what it looks like over time. And so what you see is that the demographic pressure that we have now is exactly the opposite, we would think. If you want to control population growth, you lower childhood mortality. And that's because of uncertainty around these things. So if you want to control population growth, you do three things. You educate women, you give women control over their own reproductive decisions, and you lower childhood mortality, and then you will lower that. And what I want you to see is that many of these yellows, which are mostly, again, in Western Europe, are below zero. So those are countries that are shrinking right now. And so it's really immigration pressure that is keeping those populations going. So I think if we continue on our overall social trends, that population growth is not going to be an issue. The economic one is real, though. And so what do we need to do about that? Well, I think what people get at when they tell me that they're not so wild about this is really has to do with thinking about what it means to live longer. So we really don't want to talk about lifespan, we want to talk about health span. So we don't want to just live longer, we want to live better longer, okay? So when I say to people, maybe we're going to find things that are going to make people live longer, they imagine our current health care situation, which is really about treating the end of life and then extending that. So the idea is not to make 90-year-olds live to be 120. What we would like to do is to have people who are living to be 120 who have more of the physiology of a 50-year-old. Okay, so it's really health span that matters. And so we can think about maintaining health status, and what we really want to do is shift out that health status. I think if we ask people, how would you like to live your life? What they'd really like to say is, I'd like to live as a 30-year-old until I'm 90 and then be hit by a bus, okay? And so the question is, can we achieve that? And there might be an intimate linking between health span and lifespan that we can't separate. And then the real question is, can we then really think about extending that out and more, such that we really are at a place where we do achieve this 2,000-year sort of horizon, which I think is not completely unrealistic, except for one thing. Okay, and that is the two major causes of aging are breakdown in genetic information, usually via mutation, and then an accumulation of protein damage. So for most of our tissue, mutations lead to cancer. So I think that those are solvable problems that we'll address. The accumulation of protein damage in most of our tissues is addressed by just making new tissue and then killing old cells. But there's one tissue in which this doesn't work, and that's our brain. And so those of you who have aging relatives know that dementia is really the rising issue about longevity now. And that's because our neurons don't replenish themselves. And so as they accumulate damage, they can only be around for so long. And so the real challenge for us now is to figure out how to replenish neurons, either to make them very robust or to replace those. And so I think if there's an upper barrier to longevity, it's really about the neuronal function that we have, and we really don't know how to solve that problem right now. Okay? Much like the examples that I showed before, we know how to accelerate neuronal degradation. This is Woody Guthrie. People are laughing at this machine kills fascists. So obviously a folk singer. And so Woody Guthrie, though, tragically has a disease called Huntington's disease, in which there's a defective protein that accumulates, and it leads to early onset dementia in your 50s, and it's a dominant mutation, but you don't know you have it. Now we can genetically test for it, but historically people didn't know that they had it until actually after they reproduced, and the onset was much later in life. So right now we're at the stage with neuronal diseases in the same way we're just generally about any kind of function, that we know how to make neurons weaker, and there's other genes that are associated with Alzheimer's disease, but we don't know how to make neurons live a lot longer yet. And so that's really where we are. So here's the worm. This is from a company that was founded by my colleague Sean Lockery, and so worms pump. And so they actually create an electrical signal when they pump, and it's sort of like an electrocardiogram, okay? So we can actually study the worm's nerves as they're firing and then start to think about how can we screen for things that make that nervous function work better. It turns out that worms start sort of losing their ability to eat as they get older, just like all of us, okay? And so, so this is a great example of a company that was born out of the University of Oregon that was created to solve a specific problem, and then Sean and I have collaborated to apply this in the aging field now. I just wanted to show you this one result because this is just so cool, even though it's not, it's difficult to assume. So what we can do now, technology is amazing. So what we can do now is take a single worm and break it all into its individual cells, and then put them into a machine that tells us what genes are turned on in each individual cell. It's unbelievable. We couldn't do this even 2 years ago. So if we do that, then we can detect every different cell in the worm as what they're doing. And so I just wanted to highlight the fact that we can actually now tell what the neural cells are and ask every single gene that's functional in a neuron, okay? So we can now ask worms that live longer versus those that don't. How are your neurons doing? Which genes are acting? All this sort of stuff. So this is the technological revolution that, that scientists are able to take advantage of, and that's what we really want to also accelerate as we move into the night campus. Okay, so this is going on. I had one more story to tell you, but that I'm going to skip over, and it has to do with what it means to live multiple lives, because we each experience just one life, but when we think about intervening in terms of our own health, what we really want to do is perturb the trajectory of what we're doing. And we're doing a lot of those kinds of experiments too. I'll just skip quickly to this is the many lives that you could live. So we build these things called microfluidic devices that we can, this is cool, but we'll skip it. So where we could raise each individual worm. So one of the cool things about these worms is we can have them be genetically identical. So you only live one life. And in your life, you've had many unique experiences. Let's say that, you know, it was raining one day and you ducked into a coffee shop and there happened to be a beautiful woman there, and that turns out to be your spouse. And if you hadn't forgotten your umbrella that day, then you never would have gone to the coffee shop, so on and so forth. So all of us have those little events that change us. Imagine if you could live your life again, and again, and again, and again. How many times would you come up with that same outcome? Well, imagine if we were measuring your health outcomes that way. Well, in worms, we can actually do that experiment because we can raise as many genetically identical individuals as we want. So we can put them in this Groundhog Day hell in which they just keep living the same life again and again, and we can see all the possible outcomes. And the intent here is to see if there's some kernel of oneness and truth about the way an individual is. I call it the stochastic kernel that defines the way that you are in the world as you run through it. So I just want to finish up. This is Dorothy Coleman. I just happened to run across this on the internet, as one does these days. Dorothy is 105. She lives in San Diego, so the San Diego Union Tribune did a story on her. And she's apparently still plays music for her women's group and Kiwanis Club and all those kinds of stuff. And so they asked her, what's your secret? That's what we tend to do. What's your secret? And her answer was, keep smiling. So that's really my message to you. Now, we could have found someone else whose primary message was, take a shot of whiskey every day. And this is the problem with anecdotes, right? This is the anti-science, is that you can find someone who does something. So we actually really need to do these simultaneously. But I do think that we have the potential to change our own outcomes based on the way that we are in the world, and that we're at the precipice now of really thinking about how to do that systematically. I think the answer, unfortunately, for some of us is not going to be what we're going to do at the end of life. It's going to be what we do when we're 30 years old, because we really need to change those trajectories early on, and that's where the revolution is going to come. So the revolution is not going to be, it's not going to be televised in our lifetime. It's going to be probably the next generation is really going to be affected by this, but we're really figuring this stuff out. So when we bring this back to this impact cycle that President Schill talked about, the idea is that we're making discoveries, and that gives us insight into some fundamental process. And historically, that's at the University of Oregon especially, that's where we would stop. But the idea now is to then take those out into an impact. So not only do we understand the biology of aging, but we think then how we can use that to make people live longer, especially healthier lives later in life. And that's, I forgot to tie the bow on the economic impact. That's the economic impact. It's not just that people living longer are going to have negative economic impact, but if we can make them live healthier longer, then that will reduce the impact. And then we can start thinking, if you could live to be 500 years old, how would you approach your life? How many careers would you have? What kind of education would you do? How many instruments would you learn how to play during that time? I think it'll change the way that we view ourselves, and it'll change the way that we interact with each other, and it'll change the way that society is structured. And this is coming, and we're not prepared for it. So that's why I really thank you for being here tonight to really think about these questions with me. Because I think the bottom line is that, yeah.
Can I ask a question?
No, just wait a second. That's my mother, okay? There's one rule, never bring your mother to your talk. My dear, if you'll just wait literally 2 more minutes, then we'll finish this baby off. Okay, before I was derailed, let me finish this up. So the idea is that science is going to, our life is going to be extended. So it's not really how long we live, but the life that we choose to live is where it's going to come. So this is going to happen. We're working on it. There's no stopping it. So one response would be, let's just not do this, okay? We like the way the world is. Let's not change this. That's not a realistic response to scientific progress. Instead, we should really be thinking about what does it mean? How are we going to support it? And I think in particular, we really need as a society to think about what it means to live a life of dignity, and in my own personal opinion, to empower people to have a little bit more authority over their own lives late in life. But that's a separate issue. But science will do this, but science won't tell us how to live a life of meaning, what that means, how should we love, how should we be loved. Those are not scientific questions. In order to address those things, we need the rest of culture. We need art, music, philosophy, and indeed science has a role to play there. And I like to think that the university has a central role in helping us to think about how those questions should be addressed. But it's really up to you, my community, you're my community, to think these through together and to really talk about them. Which is why I'm really happy that we had a chance to at least begin to explore this conversation, especially in sort of thinking about it in an open, rational way of addressing these questions. Because we're clearly going to need to think about this, and we really need to be thinking about the future that will be coming. And this is just one example of many of these things that are going on. Just this year, Chinese scientists genetically engineered the first human to cure a genetic disease. So if we consider aging not a good thing to do and a disease that we can cure, we could start genetically engineering. This will all be happening in the next decade. So we really need to be thinking about how we're going to approach that. So with that, I'll end, and I'll thank you very much. So it's getting a little late, but I did want to give an opportunity to answer a couple of questions, including one from my mom, potentially. But I'll just take maybe 3 questions so that the rest of you who want to get on with your evening can do so. But what I'll do is, after we're done, I'm just going to be right over here. If you want to come and chat some more, I'll stay as long as you want to keep talking. So I wonder, we have a couple of mics here. That are ready, so because I'm sure people want to hear your question. Are there questions? Let's go. Give her that woman right there. No, you have to speak into the mic. This is how it works. It's on. Just speak. Yeah, no, you just speak into it.
Am I okay now?
You're good. Go for it.
This is just too simple. If we live to be 100, are we going to keep getting more and more wrinkled every year?
Oh my God. You know, if you live in Oregon, you get no sun, and so, that is part of aging. So that's, again, we could have talked about this for hours and hours. Aging is not just one thing, it's many things. So skin elasticity is one. There's a phenomenon called sarcopenia in which we start losing muscle mass. And even if you exercise late in life, you're going to lose your muscle mass. It's really hard to control. These are all things that we need to look at. The question is, and this is really a fundamental question in the field, is there just 1 or 2 things that we can do that will start addressing all of those, and so that our skin, 30-year-old skin is still 30-year-old skin? Or are we going to have to solve each problem individually? And we don't really know the answer to that. And so this is something that the NIH is interested in. And so actually we're just about to start a new grant that is specifically focused on asking this question from a systems perspective. Are there just a couple of drivers biologically that if we tweak them, we can address the aging question? Or are we going to have to fix 10,000 things that start breaking down over time? The fact is that the trees live 4,000 years. Elephants live a long time. Tortoises. So we know that it's at least possible to maintain function that way. Again, I think the problem is really not in how wrinkly we are, but how our brains are going to function, because that's our sense of self as we move forward, and that's what we start losing. Another question go ahead.
I'm an alumni and a happy alumni at that. I'm also a CEO of a biotech company. How do we work together in this coming Impact campus in a way that keeps track of the intellectual properties which are important on the entrepreneurial side and maybe even on your side?
Yes, so that's definitely a big part of what we're interested in. Again, I didn't have time to go into this. A big part of what we're trying to do is actually support entrepreneurial activity through this. So we'll be supporting startup companies, have entrepreneurship programs that will be supporting our students and figuring out how, so they can be the next happy alumnus who started a biotech company. That's our a great ideal to have. So we already have operations in place that help protect intellectual property that comes out of the university at the same time of rewarding the people who make those discoveries. What we really need to do is sort of amp that up at a big level, and so we're working on creating structures to do that. There's a question there.
Hi, my question is just, we know there's lots of difference between worms and people,
Yeah.
but as you look at it, you've talked about keeping, as you've studied the worms, similar situations. What happens when you put them in a lot of environmental change? And do you see the same consistencies, and can you recreate them when you put them in with a lot of environmental differences?
Yeah, so that's one of the things that we found. I just gave you a brief little picture of that with the heat, that there's actually a lot of environmental inducers that will be protective. If you give it, it's a phenomenon called hormesis, where you give a little bit of something and then it upregulates. What we think biologically what's happening is they sense, oh, I'm coming into a stress, and then it'll go up. One of the fascinating things, and again, a whole nother talk, is that we've now discovered that those things are transmittable. Those stress experiences are transmittable across generations. This is something we didn't think was actually possible. And so in worms, if you starve a worm, 15 generations later, there's a genetic signal of that starvation event, for instance. And so, so this interplay between the environment and genetics is going to be really important going forward. And to some extent, the compounds that we're studying, one way of thinking about them is to create sort of an artificial environmental signal. So you're turning on responses just by taking something rather than having to, walk over hot coals or whatever. I'll take one more question. Yeah, here comes a mic.
Yeah, recently I read about telomere extensions where somebody extended their telomeres by 600 base pairs. And I was curious, will that take away with the wrinkles?
So there's lots of cellular phenomena that can lead to aging. One of them is we have these ends of our chromosomes. They're called telomeres, and those of you who are aging aficionados have probably heard of this. And so what happens in our normal, what are called somatic cells, those get shorter every single time that the cell divides, until at one point then you start chewing into the chromosome itself. And so one of the ideas on aging is, can we extend those? And the problem with that is that then you make it more likely that you'll get cancer. And so it's a balance between those things. Will that solve the wrinkle problem? I actually don't know enough. I got it, obviously, for the next talk. I'm going to study up on collagen biology. But I think that I don't think that that's going to be the essential problem there. But it is true that what happens is that we start getting dysfunction in the organization of our tissues, and some of the signaling just starts going wrong. In the case of wrinkles, it's really actually due to fat maintenance, I believe, and so that the subcutaneous fat starts going away in ways that we would not want it to. Okay, so I'm going to wrap it up. So I thank you again, and really appreciate you being here.